Hello and welcome to this introduction on gel electrophoresis. Today we're going to learn about what gel electrophoresis is, how exactly we do it in the lab, and what benefits it will have for us. Well, what exactly is gel electrophoresis and why do we want to do it? Well, what it will allow us to do is separate our DNA fragments of our experiment based on how big they are. First of all, a gel electrophoresis gel is actually about the consistency of Jell-O, give or take, and so all of the DNA fragments will be able to move through the matrix of this Jell-O. If you take your pipette tip and you slowly lower it into the well that you've created with your comb, you want to very, very slowly depress the plunger on the pipette until your sample is loaded into the well. It will not fill up the entire well. You'll load about 10 microliters or so. Then you connect the apparatus to the electrical system, and the gel will allow the DNA fragments who are attracted to a positive charge to very slowly move through the gel from the negative end to the positive end, and they will do this according to how big they are. Since we talked about the gel being a matrix, kind of a web, it will allow the smaller pieces to move through a little bit faster, and it will allow the larger pieces to take more time to move through the gel, and so they will appear closer to the beginning. First and foremost, you need to make sure that you are putting buffer inside of your sample tube. You never ever want to load a gel apparatus that doesn't have buffer with it, otherwise you will lose your DNA sample because it will float out of the well.
Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger.
The first two examples I want to show you are normal loading. Notice I put the tip down into the well. When I depress the plunger, the sample will drop down to the bottom. As I pull away, you'll also notice a small stream. If this is a worry to you, just hold your tip down there longer so the sample can drop down. Otherwise, it's usually no big deal when you're loading a sample. The next couple examples are your classic air bubbles. The first one is the air bubble at the tip. This is usually no problem. Just before you put it into the buffer or to the electrophoresis buffer, just depress your plunger to get rid of it. The second one is air bubbles within the tip. If you see this, be aware of it before you try to load it. Put it back into your tube, draw it back up without air bubbles. Air bubbles in the tip are going to cause dispersion of your sample and possibly lead to some of the loss of your sample. Here is the classic shoving your tip through the bottom of the gel. Just be aware of where your tip is. As you can see down at the bottom, the sample is lost underneath it, and also you have inadequate loading of your sample into the well. Sometimes you can save your sample or have enough in your well to actually do the electrophoresis, but other times you kind of just lost some of your sample. Sometimes when you're in a big hurry purifying your DNA, you will leave a little bit of ethanol over your pellet and then dissolve it in your appropriate buffer, usually TE buffer. If there is too much ethanol or isopropanol or some organic solvent, when you go to actually load your gel, you'll notice that your sample will actually float out of the well into the buffer. And so here's just a little video showing that. Here's a quick example of what happens when you don't put your tip into the well. As you can see, the weighting agent weighs your material down into the well, but periodically you'll see samples spilling onto the surface. This is usually not a huge deal, but it can contaminate local wells, which could lead to false readings. So just be concerned of where your tip is and try to put it a little bit into the well to ensure proper loading. Last but not least, here's a quick example of what happens when you do not remove the tip before you pull the plunger back up. You'll load some of your sample back into the tip. This actually may not be a big deal, but if you're trying to be quantitative, it may lead to false results.